Today we are going to discuss interferons. Okay. Before, we have discussed about bacterial infections and how the cells behave when there is some bacterial infections, like how the cells detect bacterial infections and how the cells are going to respond to bacterial infections. The bacterial infections are mostly extracellular, and you have gone through that stuff in detail, that how the toll-like receptors, whether these are endosomal toll-like receptors or toll-like receptors present on the surface of the membranes, these receptors recognize different kind of bacterial pathogens, and then they would lead to this cascade of events inside the cell, finally leading to the transcription of genes that are involved in the process of inflammation. Okay. Now there are viruses as well, and you know that the viruses, they cause intracellular infections. They would just enter into the cell. So how the cells behave when they are infected with viruses? The cell, actually all nucleated cells in our bodies, they are prone to viral infections. They are susceptible to be infected with different kind of viruses. And all these nucleated cells have their response in this case when they are infected with viruses. And that response is in the form of secretion of interferons. Okay, so this is the natural antiviral defense of the cell, that the cell is going to secrete interferons when the virus infects that particular cell. Interferon receptor is constitutively expressed on the surface of all cells, okay, all nucleated cells. So the cell doesn't need any kind of induction for expression of different receptors. As you would have seen in different cases, that some of the receptors, they are expressed when the genes responsible for making that particular peptide receptor are induced by some mechanism. But these are constitutively expressed, which means that they are made as part of the cell when the cell is formed. Now, what is the action of interferons? The interferons, they interfere with the viral replication. That's why generally they are known as interferons, And they also alert the immune system about infection, that there is some kind of infection, okay? Thus causing the virus-infected cells to become target of the natural killer cells. The natural killer cells are naturally equipped with this capability to figure out that what are the cells or which type of cells are infected with viruses, because viruses are hidden inside the cells, okay? So if it were bacteria or other pathogens, they are present outside the cell. So it's easy for the cells to detect these pathogens with the help of different receptors. But viruses are inside, they are intracellular, okay? That's why there should be some mechanism for differentiating between an infected cell from a non-infected cell. So the natural killer cells have got this capability naturally. That's why we call them natural killer cells, They can do that particular job. So interferons also help with, this killing of the infected cells via natural killer cells, and interferons directly can interfere with the replication of the viruses to stop the virus from propagating inside the cells, okay? So you would have seen that there are lots of different interferon preparations available on the market, and people have been using these interferon preparations in case of different viral diseases. For instance, in hepatitis C virus infections or hepatitis B virus infection, different types of interferons, alpha or beta interferons, are being used. They are synthesized by different companies, and they are marketed to treat these infections, in different people. Okay, so why? Because they can stop the replication of these different kind of viruses. So they can directly stop it. They can induce a number of genes that we call interferon stimulatory genes. And those genes can make products in order to stop the replication of virus or the transcription of virus or to prevent the assembly of different proteins that are made inside the cell. Because the virus once enters the cell, it is going to hijack the entire machinery of the cell for its own purposes, and the purpose is to replicate its RNA or its DNA and to make its own proteins. Because the viral genome is so small, it doesn't have a number of enzymes required for this particular task, for these different types of tasks. That's why the virus is going to take benefit of the cellular machinery, the eukaryotic cellular machinery, to make its own products and make viral particles, So these interferons, there are a number of them. It's a whole family of genes of interferons that we are talking about. And this family of genes is located on chromosome 9, okay? So this family includes many different types of interferons that you see in red here. Interferon alpha, beta, kappa, and omega, and theta, gamma. All these are different types of interferons. We also call them type 1 interferons. Okay? And these type 1 interferons, they act like cytokines. Different types of cytokines like interleukins or tumor necrosis factors that you have studied. The interferons also act like cytokines, Cytokines are short-lived. Interferons are also short-lived. The cytokines are short-acting. Interferons are also short-acting, When cells produce cytokines, these cytokines are not there to live a longer life, just for a short period of time. For instance, in the case of infection, for a certain period of time, until and unless that infection is cleared. And these cytokines are not there to travel longer distances, These cytokines also, interferons also, just like the cytokines, can act via autocrine mechanisms or paracrine mechanisms. Autocrine mechanism is that interferon is produced by a cell, secreted from that particular cell, and acts back on the same cell, okay? Or it influences or stimulates the same cell that has secreted it. So this kind of action is seen in the case of different cytokines, and it is true in the case of interferons as well. And then there is the paracrine mode, in which interferon is secreted by one cell, and it is going to act upon another cell. We call it a paracrine mechanisms. So again, this is true for both cytokines and the interferons. Okay, they act like that. I will also show you some images in the next slide, so it would become easier to understand. Now, as I told you that all the nucleated cells in your body, they can be infected with viruses, and any cell that is infected with virus is going to produce interferons. Okay, so it means that all nucleated cells can produce these interferons, in situations when they are infected with viruses. But there are some specialized cells like this one, plasmacytoid dendritic cells, or PDCs. These cells look like this, as you see here. This is a cell. It looks like more, a B cell, a plasma B cells, for instance, which makes a particular type of antibodies, So there is extensive endoplasmic reticulum if you just look at this image. And this kind of these plasmacytoid dendritic cells are specialized cells or factories for the production of interferons. They produced 1,000-fold more interferons than all other nucleated cells. Okay? So that's why these are specialized cells. You know about these dendritic cells. Dendritic cells are present in the tissues, just like the macrophages. And when they come in contact with antigens, these are antigen-presenting cells, they would take those antigens and go inside the lymph nodes and present these antigens to the T cells. You know about that, okay? So there are different types of dendritic cells. One type is the myeloid, dendritic cells. We call it MDCs, okay? And the other one are plasmacytoid dendritic cells, that are these cells. So these cells are for the production of interferons, while the myeloid, dendritic cells, they are doing antigen presentation as a typical job of the dendritic cells. Okay, so that is a different type of dendritic cells from this one. This is more like a B cell. The B cell is producing a single antibody, and this cell is producing a single peptide, which is interferon. All, of course, many different types of peptides, but all these are interferons. Okay, so what is there inside these cells? That since the viral infections, they express toll-like receptor 7, for instance. These cells, they have toll-like receptor 7, and they have toll-like receptor 9. And these toll-like receptors are endosomal. They are present inside the cells or in the cytoplasm. Okay? So toll-like receptor 7 recognizes single-stranded viral RNA, because the viral RNA, in case of RNA viruses, the genome is, maybe single-stranded. During their process of replication, it can encounter single-stranded RNA and bind to the single-stranded RNA. And also the C plus G islands. And I also have told you about this, that the C plus G islands are more frequent in microbial genomes as compared to the human genome. Okay? That's why inside a human cell, this receptor can specifically bind to microbial nucleic acids and not the nucleic acids of the human origin, thus differentiating microbes from the human DNA. Okay? So what happens when these receptors would bind to their respective ligands, these RNAs or DNA molecules of the microbial origin? Now, the signaling that is going to take place is the same as we have studied before. It would be, the action would be through the MYD88. Okay, you have studied about that. That finally is going to lead to NFK-beta, activation of NFK-beta or release of NFK-beta, or the interferon response factor 3. Okay, and these factors are going to enter the nucleus. They are translocated into the nucleus, and inside the nucleus they would start mass production of these interferons. Okay, you understand? The signaling that you have already studied in case of bacterial infection, for instance. So there is a little bit difference, but finally, when the signals are passed through adapter proteins to the through adapter proteins in the form of phosphorylation and dephosphorylation of different molecules, a cascade of events that would just ensue when these receptors would find these RNA or DNA of viruses. So finally, it's going to lead to the activation or release of NFK-beta. which is a transcription factor, and interferon response factor 3. Again, it's a transcription factor. So where are the transcription factors destined to go? They are going to be translocated into the nucleus. They enter into the nucleus because transcription factors act on different genes. They activate different genes. Okay? So NF-beta and IRF3, they would just act upon different types of interferon genes that are present on chromosome 9. And they would activate all of these genes to start production of interferons, mass production of interferons. Okay? It means that NFKB and IRF3 are specific transcription factors for the activation of all these genes in plasmacytoid cells. Okay? Specific transcription factors or enhancers of transcription are different for different genes. And in different tissues, there are different kind of transcription factors available. We already have studied that. In plasmacytoid cells, these are the transcription factors responsible for starting the transcription of all these genes. Okay? Now what happens, why is it needed that the plasmacytoid cell should produce interferons in such, a large quantity? And that quantity is required in order to prevent or stop, the chances of, or minimize the chances of systemic infections. That's why when interferons are produced in infected tissues by these cells, then all these interferons, they are, transported to different parts of the body through lymph and blood. Okay, so these are, circulating blood or lymph. These are going to different parts of your body. In taking these interferons with them. So what would happen when interferons would reach to different parts of your body through, these routes? Then the chances of systemic infections will be minimized in that case. Okay? So that's why these cells are called as the factories of interferons.